Hi, I'm Shirley Tsunoda, and I'd like to tell you about our projects for the last year around pharmacomicrobiomics and metabolomics. And I run the data science project together with Peter Dorenstein. So we are interested in the mother-infant dyad and the intersection of the communication between them, including HMOs, the microbiome, and the metabolome. And we're particularly interested in the exogenous influences, such as drugs, food, and other environmental chemicals that a mother and baby may be exposed to, such as personal care products. We've established many analytical and computational tools in order to study these questions. One of those is a drug library. So, as many of you may know, when we're doing a metabolomics experiment, it's sometimes difficult to annotate the millions, thousands of chemicals that are present in a given sample. And we are particularly interested in measuring drugs and drug metabolites, including microbially mediated metabolites of these drugs. And so Nina Zhao, one of our talented postdocs in the group, has created a mass spectrometry drug library in order to improve the annotation of drugs and drug metabolites in our samples. So she took publicly available data and organized this into a library of almost 5,000 drugs, and these also include analogs and metabolites. And Kine helped her by providing the metadata associated with those drugs. So, providing the source of where that drug came from, the disease area that those drugs are mostly used in, the pharmacological class, and the therapeutic indication. And I hope you'll watch Nina's talk. She will be talking about the drug library as well as another project in breast milk. So this is the project that Nina is going to provide more details on in her trainee talk. We had the fortune uh, to have the ability to annotate drug detections in about 2,500 breast milk samples that were collected from our UCSD Human Milk Biorepository that's run by Tina Chambers. And what we found was that, probably not surprisingly, antibiotics are one of the largest class of drugs found in these breast milk samples that have been collected over about 10 years or so, and followed by antihistamines, GI drugs, drugs for mental health, such as antidepressants, and then, a smattering of other drugs in different categories. And Nina's going to talk more in her talk about our analysis on the metabolome of breast milk when these drugs are present. Simone Zuffa has conducted an animal study looking at the impact of perinatal ampicillin on the maternal metabolome and microbiome. And he's also giving a talk, a trainee talk, to describe this project in more detail. So I'm just going to highlight for you what he did. So, in collaboration with the Nizay and Liu lab, we subjected mice to both antepartum administered ampicillin and postpartum administered ampicillin. So, antepartum sort of mimicking the prophylaxis that mothers get for GBS, and then the postpartum administration of ampicillin reflecting just, ampicillin being given after the baby's born. And stool was collected and subjected to untargeted metabolomics and shallow shotgun metagenomics. And not surprisingly, we found that ampicillin reduces many different bacterial species, and most notably, many beneficial bacteria that are short chain fatty acid producers. So, 32 species showed a concordant reduction in both the antepartum and postpartum administration with ampicillin, while enrichment of bacteria, that is, bacteria that grew in response to ampicillin, seemed to be cohort dependent. And some of those SFCA producers include Muribaculum and Akkermansia, Prevotella, and Eubacteria species. Interestingly, Simone found that antepartum exposure actually had a longer prolonged effect compared to postpartum ampicillin administration. And this was shown both for the microbiome and the metabolome, with this effect being seen until weaning or postnatal day 21. Whereas postpartum ampicillin demonstrated alterations in the microbiome and metabolome during administration, but seemed to recover by postnatal day 14. And then we looked at the bile acid distribution and what happened to bile acids in response to ampicillin. And we found that in the antepartum period, secondary bile acids were depleted, whereas primary bile acids were enriched. And the same was true for the postpartum period, but there was an earlier recovery of those bile acids. There were more metabolic features altered with ampicillin when given in the antepartum period compared to the postpartum period. Other metabolic features that were altered included carnitines, polyamines, and small peptides. And as you probably remember, secondary bile acids do play a role in signaling and in regulation of the immune system and lipid pathways. So the fact that those were depleted with ampicillin administration suggests that some of those pathways could be disrupted. Kine did a very interesting study in collaboration with Janet Markle at Vanderbilt, and this was recently just submitted for publication. So Dr. Markle has a very interesting cohort of very early onset IBD patients, and she gave us fecal samples from 26 VEO IBD patients and 28 controls. There were 54 fecal samples, and we subjected them to microbiome analysis by 16S and untargeted metabolomics. And then Kine processed the data through Cheetah, MZMine, and utilized GNPS to identify the metabolites. And Kine also has a presentation in the trainee session, so I encourage you to view her presentation to get more details. I'm just going to give you the highlights. So, untargeted metabolomics identified a depletion of short chain N-acyllipids and an enrichment of dipeptides, tripeptides, and oxo bile acids in the VEO IBD patients. And then, interestingly, there was an alteration of several different bacterial genera in patients with VEO IBD compared to their healthy controls. So there was a lower abundance of beneficial bacteria such as Bifidobacterium and Blauschia, and a higher abundance of Lachnospira, Veillonella, and Bacteroides in VEO IBD. And then Kine did a multiomics analysis of the metabolome and the microbiome and basically showed a connection between N-acylipid production, which was positively correlated with Bifidobacteria, Blauschia, Coprococcus, Collinsella, and Doria. And these genera were more abundant in the healthy. And then she found that the N-acyllipids were negatively correlated with other bacterial species such as Lachnospira, Bacteroides, and Allostipes. So Kine also, we also had the pleasure to be involved in a collaboration with the Gates Foundation, and Kine worked very hard on this data. And this was a cohort of women and children, 55 mother-infant dyads that were recruited and enrolled in a study that was done in Dhaka, Bangladesh. This is an observational study over the first 6 months of an infant's life. And we had lots of samples that were collected from these patients, from these participants. plasma, fecal, breast milk, infant tongue scrapings, infant skin, and vaginal fluid. And HMO analysis was done, untargeted metabolomics, and microbiome analysis, shotgun metagenomics, was done on all these samples. And she conducted the metabolomic analysis, and the microbiome analysis was done by postdocs in the night lab. And what we found was that there was a unique microbiome trajectory at each infant body site, dominated by environmental factors over maternal sources, with safe water emerging as a critical factor. And I'm just showing you here the milk metabolome data. And we found it to be very interesting that there were some external factors as well as some internal factors that influenced how the metabolome developed over time. So, we were able to see a trajectory over time because these infants were sampled over 6 months period of time. And we saw that the metabolome developed over time and that it was influenced by delivery mode, so whether or not the infant was delivered by C-section or vaginally, the mother's HMO secretor status, so whether they were secretors or non-secretors, and the mother's exposure to filtered water. We also saw that there were bile acid profiles that changed with age and that were associated with HMO status. So this study provides some insight into the development of the microbiome and the metabolome in a developing country. And finally, I want to tell you about our clinical study that we have run for the last several years, headed up by Tina Chambers. She and her team enrolled 80 mother-infant dyads, and we had 40 mothers who were exposed to antibiotics and 40 who were not. And we collected fecal samples from the mother and the infant, skin samples from the mother and the infant, breast milk samples, and environmental swab samples of the environment of the home of the mother and baby. So all of these samples have now been collected, and we've run them all on our LC-MS/MS platform for untargeted metabolomics analysis. And Kine is going to be very busy analyzing these samples and trying to answer some of these questions. For example, what is the impact of antibiotics on the infant microbiome and metabolome? What is the role of the mother-infant dyad in the environment, the mother-infant dyad environment in transferring antibiotic effects? And what is the impact of antibiotics on infant growth and development? Additional analyses that we're doing include targeted metabolomics by Jeremiah Momper in our pharmacometrics core, HMO analysis by Lars Bode in our milk analytics core, and microbiome analysis of the fecal samples in the Rob Knight lab. So to summarize, we are very interested in this trajectory of pregnancy, delivery, infancy, and toddler age, because this is a very critical time of the microbiome being developed, the immune system being developed, and obviously very many developmental milestones happening during this period of time. And we're particularly interested in drugs, not just antibiotics definitely, but there are many other drugs that we know impact the microbiome. And how does this impact the eventual development of the microbiome and then downstream health and development of the infant? And so at any period of this developmental timeline, drugs, whether it be antibiotics, antidepressants, other drugs that the mother or the baby may take, how does this impact this trajectory? And we use the tools of metabolomics, microbiome analysis, and computational strategies to try to answer some of these questions and hopefully give us some insight into preserving and optimizing the health of our children. Thank you so much for your attention, and I look forward to discussion.